The next carbon compound, macromolecule, that we're going to talk about is proteins. And proteins are incredibly important inside the body, as are all the macromolecules, but proteins basically do everything that your body does. Every job inside your body is carried out by a protein. And if we were to remove all of the water from our bodies and only have molecules left in compounds, proteins would make up half of what's left after we removed water. So that's huge. Proteins are by far the most abundant kind of molecule inside our bodies next to water. And they do everything, like I said. So here's some examples of what proteins do. So they are enzymes, which means they make reactions happen. They store things, so they can store amino acids, especially like in growing embryos, store them for the growing embryo. They can be hormones to regulate your body's activities. They are involved in movement. This is probably what you guys think about when you think of protein. You don't think of the kind of protein that stores albumin for a growing egg embryo, but you think of your muscles, which is a huge part of what proteins do, movement. We have defensive proteins, which protect us against disease. They recognize and start an immune response against invaders. We have transport proteins, which allow for transport across cell membranes. They act as little tunnels or trains to move proteins, to move other molecules. We have receptor proteins, which act as chemical messengers. They sit on the outside of cells and receive messages. We have structural proteins, which help in support. So collagen is a very big component of our skin that gives it its elasticity. So those are the many different kinds of proteins, but basically enzymes are one of the most important. Life couldn't exist without enzymes. So enzymes are molecules that allow reactions to happen. Basically, chemical reactions would take so long on their own if they didn't have enzymes that life couldn't happen. But enzymes speed up reactions and allow them to happen. And the key thing about enzymes is that they don't get used up in a reaction. They are unchanged at the end of a reaction, so they can do their job over and over and over and over again. So your book calls them the workhorses of the cell. They carry out the processes of life. So getting into the structure of proteins. Proteins are made up of what we call polypeptides. Poly meaning many, peptide meaning the kind of bonding that proteins do, and we'll see that. They are unbranched polymers, so they're a straight line made up of many, many amino acids. There's only 20 kinds of amino acids, but think of how many words can be made up from the 26 letters in the English language, and that only goes to maybe a dozen letters, whereas polypeptides go to thousands of amino acids. We can make crazy amounts of different kinds of polypeptides. And so polypeptide is just like this straight line of amino acids. Then it will fold up and bind with other peptides to make a protein, and then the protein can do its job. So a protein is technically the biologically functional molecule that consists of one or more polypeptides. So the polypeptides are built, and then they fold up, and then they can do their job. And they're made up of amino acids. So amino acids are organic molecules that are characterized by their structure. And all amino acids have a carbon that's bonded to 4 different things. It's bonded to a carboxyl group right here. It's bonded to a hydrogen, and it's bonded to an amino group. And then this R group over here is what's different between each amino acid. So each amino acid in the whole entire world has a carboxyl, a hydrogen, and an amino. But this R group is what's different between all of them. And the R group is what gives the amino acid its different properties. So we have some polar amino acids, some nonpolar, some acidic, some basic, and that's all from this R group right here. So here are all the different amino acids. Here are the nonpolar amino acids based on their side chains. They have nonpolar R groups. So you can see, we see a lot of carbons and hydrogens, which means there's no polarity. It's nonpolar, which means they do not blend well with water. They are hydrophobic. Then we have the polar side chains, which lend to polar properties, such as being hydrophilic. You can see a lot of oxygens, double bonds. That's what leads to the polarity, the unequal sharing. Then we have some electrically charged side chains, which the negative ones are acidic and the positive ones are basic. And these are hydrophilic because any charge attracts water. So the different R groups are what give the amino acids their properties, because the other 3 groups, the amino group, the hydrogen, and the carboxyl, are all the same on all amino acids. And so these amino acids are linked by peptide bonds. And so a peptide bond forms between 2 amino acids what happens is when the carboxyl group of one amino acid lines up next to the amino group of a second, they come together. The hydrogen of the carboxyl group, the hydroxide of the carboxyl group bonds with the hydrogen of the amino group. They leave as a water molecule, and a bond is formed through dehydration synthesis. And this kind of bond is called a peptide bond. So if we have many peptide bonds, we have a polypeptide. And so these bonds actually have directionality. So that means that we can talk about one end of a protein as being different from the other end. And that is characterized by the end that has the amino group sticking off. We call it the N-terminus because there's a nitrogen. And the other end is the C-terminus where the carboxyl group is because of the carbon. So that's how we can talk about the different ends of the polypeptide. And so these could be from just a few amino acids in length to thousands and thousands and thousands for each polypeptide. So each are different based on the function that it's trying to perform. And so once this polypeptide is completed, it's going to fold in on itself. So rather than being a straight line, it's going to fold in and look something like this. We're going to talk a lot about protein structure and how this happens. But the way that it folds is based on the sequence of amino acids. Because think about it, when it folds up, the polar amino acids are going to want to be by the polar ones. The non-polar ones are not going to want to be. And if it's going to be in water, the non-polar ones are going to want to be on the inside, etc., etc. So the structure of the amino acid is dependent directly on, the structure of the protein is dependent directly upon the sequence of amino acids in the polypeptide. And its structure determines its function, as we will see over and over and over again in this course. So a protein is going to be binding with something, and it can only bind to something very specific. We call it a lock and key model. Proteins usually only bind with one other thing, and it's very, very, very specific. So right here we see this antibody protein. So this is a protein inside your cell that's going to send up a flag if there's a bad invader inside your body, and this is a protein from a flu virus. So something that we do not want. Obviously, you don't want the flu inside your body. And you can see how perfectly they fit together. Structure, perfectly fitting function here. So this antibody protein is only going to bind with this flu virus, which is why when there's new flu diseases each year, it can be really dangerous because our bodies don't have the antibodies. We have the antibodies for a different flu strain. And we're going to talk about protein structure later, so that's all you need to know about proteins.